So today we will discuss about various molecular markers. Here comes the various molecular markers. RFLP, that is restriction fragment length polymorphism. RAPD is random amplified polymorphic DNA. DAF, DNA amplification fingerprinting. STS, sequence stacked sites. Also it is CAPS, cleaved amplified polymorphic sequence. Then comes SSR, that is simple sequence repeats. AFLP, amplified fragment length polymorphism. And then comes SNP, that is single nucleotide polymorphism. It is very much sure that you will get 1 or 2 questions from this topic. It's a very important topic, and to have a clear-cut idea, I have made the table upon it, and it's from the table only you'll get a clear-cut idea about each and every marker. What is the principle behind every marker? It's given in this. For first comes restriction fragment length polymorphism. Here the principle behind it is, it's the endonuclease restriction. Then comes the hybridization, that is southern blot hybridization. For RAPD, that is DNA amplification with random primers. Then DAF, that is DNA amplification with random primers. Sequence stacked sites, CAPS, that is endonuclease restriction of PCR products. First the PCR is done, then the amplified products, they are restricted by the endonuclease enzymes. Then comes SSR, that is amplification of simple sequence repeats using specific primers. See, here you know very well, in RAPD and DAF we have mentioned random primers, and in SSR, that is using the specific primers. This is very important for you to note, because in SSR we need to know the sequence to design the primers. Then comes AFLP, that is endonuclease restriction. Then use of adapters and selective primers. In SNP case, here we use the DNA chips, and then it is sequence analysis. Further comes the number of loci detected by each of these markers. In case of RFLP, the number of loci detected is 1 to 5. RAPD 1 to 10. This question is very much asked, this number of loci detected. In RAPD case and AFLP case, it has been asked in the previous papers also. So you need to note it down and clearly be very clear in this thing to mark the options. In case of DAF, that is 20 to 30. STS, CAPS, it's 1 to 4. SSR, 1 to 3. And in case of AFLP, it's greater than 70. You should note it down. It's greater than 70. Okay. Then in SNP, it's 1 biallelic. This is also asked many times. So next question, it's a very important question for all the markers. Inheritance, whether it's codominant marker or dominant marker, it should be very clear to you. So in dominant markers we take RAPD, DAF and AFLP. You make a separate note on it. What are the dominant markers and what are the codominant markers? Codominant markers comes with RFLP, STS caps, SSR and SNP. With codominant markers, you can differentiate between the heterozygotes and homozygotes clearly. Then comes type of polymorphism detected. In RFLP is base changes, deletion, duplication. In RAPD, base changes, DAF is also base changes. Same is the case with STS caps, that is base changes. In case of SSR, that is variation in length of repeats. You need to note this down. AFLP, it's base changes. Then SNP, it's single base changes. Note it down, single base changes. Then reproducibility. RFLP, its reproducibility is high. RAPD, it's very low. DAF again low. Rest of the markers, reproducibility is high. So only in case of RAPD and DAF, it's low. Rest, it's high. Then comes sequence information needed. So here are the three markers for which we need the information. That is STS caps, SSR and SNP. Please note it down. It's very much asked in the paper. In previous years, it's always asked. You need to have a clear-cut idea about this. So, and in case of rest of the markers, we do not need any sequence information for designing a primer for its use. So, RFLP, RAPD, DAF and AFLP, they all fall into the same class where the sequence information is not needed. But in case of STS, SSR and SNP, sequence information is needed. So, then comes automation. Automation, it's good in case of SNP, SSR and AFLP plus SNP. So 4 falls into this category, that is STS caps, SSR, AFLP and SNP, and rest of the 3 falls into another category. Then comes the cost. For RFLP, it's medium, the cost is medium. Then RAPD, it's low. DAF, it's medium. STS, it's medium to high. Then SSR, it's medium. AFLP is high and SNP is also high. Then comes technical difficulties. The technical difficulties in case of RFLP, DAF, it's medium and it's low in case of RAPD, STS and SSR. And rest of the 2 markers, that is AFLP and SNP, it's medium or either it is high. So, then comes the amplification. Varietal fingerprinting. RFLP, yes. RAPD, yes. DAF, of course, yes. Then STS caps, it's yes. SSR, yes. AFLP, yes. And SNP, yes. It's positive for all the cases. Then comes QTL mapping. Whether all these markers can be used for QTL mapping. It's yes. All of these can be used for QTL mapping as well as varietal fingerprinting. Genetic diversity. It's again yes for all the markers. We can use all of these markers in case of genetic diversity as well. Marker assisted selection. See, this is very important. This question is also asked. What all markers can be used for this case? That is RFLP. RAPD, it's case of yes as well as no, because it's a dominant marker. So it will not be able to differentiate between the heterozygotes. So you will be getting somewhat a bit of confusing results sometimes. So it can be used as well, and rest of the markers can also be used except for DAF. Then is qualitative gene tagging. All of these can be used for qualitative gene tagging as well.